Okay, so welcome everybody for our first immuno-webinars of this first semester of 2025. So it's a pleasure for us to receive Dr. Tiago Mineo. Dr. Tiago Mineo is also a host with us here from Federal University of Uberlândia. He's going to present his data. Dr. Mineo is an old friend of mine, and he has always worked on Toxo and Neospora caninum infection, right? Since when I know him, at least. But he has an extensive background on these two important parasites. Dr. Tiago Mineo is a veterinary doctor by formation, but has a PhD in pathology, right, Tiago? And today is a full professor at Federal University of Uberlândia. So welcome, Tiago, and the floor is yours.
Diego, thanks a lot for the nice introduction, and also I'd like to thank Gustavo and all the participants in this initiative that we've been taking part since last semester, right? I think for our program here in Uberlândia, it's very nice to be sharing all this with you. I think for the students, it's great, right? So they can hear about different topics of immunology from people all around the world. So it's been very nice for us. Thanks a lot. And also I'll have the opportunity today to show a little bit on what we do in the lab. Let me just share here screen. Just a sec. All right, so you'll see the slides. Yep. Okay, so I'm gonna tell you guys a little bit of what we do, on sort of how we started and where we're trying to head, right? And I hope this is not tedious for you guys. I'll do a little bit of introduction, but I think necessary since it's so different on my background and what we do nowadays. So I think it's worth it. So we first we work with these two parasites, these protozoan parasites. They're intracellular parasites, protozoan, and they were basically the same parasite 20 million years ago. They diverged, and they're very close together. The genomes are very close to each other, but they have a lot of biological features that are different, especially regarding the host, right? So that's what we're interested in looking at. Toxoplasma, I think it's a parasite that most of you guys know. It has a lot of seminal work published in Nature, like Discover. There's even a Toxoplasma punk rock band in Germany named after the parasite. So it's a famous parasite, right? Differently from Neospora. Neospora is shed. It was discovered in the late 1980s, and it has the dog as its definitive host. It sheds the parasites in the feces, and these parasites are in the environment, and they may contaminate food sources or water sources for the intermediate hosts, especially other dogs or ruminants or other types of animals, right? You don't see humans here because, as far as we know, this parasite does not infect successfully humans. And that's the main difference from toxo, and that's why we are also interested in it, right? How come two very close parasites that share, I don't know, around 80%, 70 or 80% of the genome are so different in virulence towards human, right? Neospora in dogs specifically, it causes a neuromuscular disorder. The dogs lose control of their hind legs. They're not able to walk, okay? And this is usually not reversible. It may also cause huge economic problems in cattle raising. This is a fetus mummified inside the cow. There are a lot of abortions. So it costs us as a country a lot of $1 billion, right? If we think about both parasites, Toxoplasma and Neospora, when we use like experimental models in mice, we see the same pattern. So this is a mice that was infected by intraperitoneal roots with actually Toxoplasma, little amounts of Toxoplasma, and then over the days, this parasite multiplies in the sites in the abdomen, and then will start spreading toward lungs, the heart, and in the end, after around 30 days, it gets chronified, especially in the central nervous system, where this mice is going to be infected for the rest of his life, right? So if we treat mice in this acute phase during the proliferation stage with sulfa, which is an effective drug against the parasites, it does reduce or accelerate this chronification, the chronification of the disease, but it does not eliminate the parasite, right? And it's always stored or at a latent stage in the central nervous system. This is also true for humans. And in the 80s, it got very important because especially HIV. A lot of people that had chronic infection by Toxoplasma and got AIDS or HIV, these parasites would do the other way around, right? They would come out of the latent stage and repopulate the organs and kill the patient, right? So that's why Toxoplasma is very important. It's one of the reasons, and it's something that Neospora does the same trend, but not in humans. Right. Let's skip this. We actually been working a lot. Diego told you guys that since he knew me, right, I already studied Neospora and Toxoplasma, and it's actually true. I started working with both of those parasites when I was in vet school in the 90s. And we started with basic stuff, just trying to do like seroepidemiological studies to see if Neospora was in the dog populations at that time. We also looked at wild animals like wolves, right? I'm a vet, so that's why what we tried to do at the time. And we also looked into different populations, human populations, right? And we did see some seropositives, unique seropositives to the parasite, but there's no reported disease in Neospora in humans described to date. Okay. Well, during my PhD, my graduate studies, I was I was at a department of pathology at UNESP, and we worked we looked at different aspects of the disease, different models. We infected eggs, embryonated eggs, to study Neospora. We did induce some lesions with that kind of model. We also did some crossovers, infection crossovers with cows infected, infecting with their meat, infecting dogs, right? And we wanted to see how dogs responded on a natural cycle when they eliminated oocysts in the feces. And interestingly, we did look for features related to immune response on those dogs that were infected. And very interestingly, it seems that when the dog is shedding the parasites in its feces, it does not actually respond, right? We only see responses by T cells in these dogs after 60 days, 2 months of the infection. We started seeing number of lymphocytes going up. It was the same thing regarding cytokines. A lot of the anti-inflammatory cytokines were up in the first days, and down, and the pro-inflammatory cytokines were down, and we had this change in profile only around 45 to 60 days, right? The same happened also with antibodies. We did not see a lot of specific antibodies in the beginning of the infection, and we looked at different subclasses of IgG, the different classes of antibodies, and they all mimic sort of the same profile. Although the recognition of these antibodies were not great also in the beginning of the infection. So everything led us to believe that this parasite was not quite infecting these dogs, but when we looked at especially their tissues after the dog's tissues after I don't know, around a year and a half after the infection, the parasite was still in the tissues and most strikingly still in the gut. So actually the parasite is able to silence the immune system of these dogs, and they're not able to respond, right? Well, then it came a turn in my career when I ended my PhD with that kind of work. We had a problem here that we could not go a lot forward with those immunology studies. Studying immunology in dogs is very hard, right? I guess most of you guys here nowadays would not even approve for dogs to be used as models. The society has changed, and the number of tools we have for dogs is not the greatest, right? So after my PhD, I went to work at USP, at the Faculdade de Medicina de Ribeirão Preto, when I met Diego to work with João Santana, and started working with the mouse model and some transgenics, nowadays transgenic parasites or transgenic cells that we use CRISPR to make. So that's what I'm going to be talking to you. From what we've done since I started to work with these models and where we're heading right now. We don't want to do transgenics, but not like freaks or transgender as our president of the U.S. has been confusing himself with. We just wanted to do some nice studies at first to see the innate receptors, how they play their parts with Neospora, and to go from there on, right? Basically, the immune response against Neospora and Toxo, it's a Th1 based response with a lot of relevance to gamma interferon in this equation. And it's really dependent on innate recognition of the parasite. We know that TLR 11 and 12 are TLRs that are basically designed by evolution, so mice can recognize Toxoplasma, which mice are a natural host of Toxoplasma in nature. This TLR recognition triggers IL-12 by antigen-presenting cells that induces gamma, that triggers all those mechanisms that you guys all know based on nitric oxides or GTPases that actually control the parasites, right? So it's a Th1 prototypic response. Of course, TLR recognition or innate recognition can get messy, but we're going to try to mess it up, right? First, regarding TLRs, the first work we did with the transgenic mice is we wanted to do basic stuff and just check if the innate pathways that we thought were acting against Neospora, if they were actually in place. So we first took a look at the TLRs, the common TLRs, and MYD88. We saw the MYD88 mice had a ton of parasites growing in their tissues. The mice were very susceptible. Without MYD88 or TLR2, IL-12 production was compromised in this mice in vivo. And we knew that when we replenished these mice, MYD88 mice, with IL-12, we would rescue them with IL-12 or interferon gamma, just proving as a proof of concept that the pathways were working as expected. We went on to check on those major players. It might seem strange for most of you guys that we were doing this kind of work with Neospora. One might guess that someone had done it before, and actually no, that there was no work to prove like nitric oxide was really important for Neospora, or MYD88 was actually inducing IL-12. So we had to do like basic stuff in the beginning, right? We went on to see nitric oxide production, as expected, was a lot produced in vivo or in vitro against the infection by the parasite. And of course, when in mice that were deficient in inos, we actually, they were very susceptible. They would die. That would be mimicked, right, by blockage by chemical immunoglobulin. These mice also had a lot more inflammation than the wild type mice in the brain. So we see here there's some inflammation to control the parasite in the brain around 30 days of infection, and the deficient mice, they would get a lot more parasite and a lot more inflammation because they didn't have the effector molecule to control it. We also saw later on that TLRs and MYD88, they actually play a role also in antibody production against Neospora. It seems to regulate the presence of those innate receptors, actually regulate the production of IgM and IgG antibodies. And we were actually did analyze the whole Th1 pathway from TLR2 to META88 to INOS, and it seems to change all the antibody production, which for us was very interesting, right? Our lab is heavily focused on antibodies and immunoassays, and this helped us to understand that other side of things. Antigen recognition was very bad also in these knockout mice. As for TNF, we saw the exact same effect. The mice knockout for TNF, they had really low IgG1 recognition of the antigens, for example, right? In the end, regarding the TLRs, I wanted to show you here that this is not all in vain. We actually think that understanding these pathways, we can modulate them and make mice respond, or the animals respond better to vaccination, for example, trying to do an immunomodulation regarding triggering that pathway, then we know it works. So we did one set of experiments using PAM3, a known TLR2 agonist, as an adjuvant in a vaccine preparation, and to check if this would help, right? Protection against the parasites. So what we saw is that, yes, if we mix PAM3 with soluble antigens of the parasites, we get increased response by macrophages producing IL-12, and that's, of course, dependent on TLR2 and MYD88. This is also true for MHC overexpression or co-stimulation with B7. When we did immunize these mice, we saw a lot of proliferation of T cells or splenocytes compared for those animals that were immunized with PAM3 as an adjuvant, and it was comparable to those immunized with Freund's adjuvant. And of course, when we challenged these mice, what we got is like a completely reduction, absolute reduction of parasite burden compared to untreated mice or mice treated only with an antigen, right? So this brings us to a conclusion that it's worth to take a look at those pathways and try to extract that knowledge towards products that can be made, right, to protect mice. All right, so we also looked at other innate receptors. We did look into nod-like receptors. You guys all know or all have heard already about the inflammasome complex. We did take a poke at that. The parasite induces a lot of IL-1 beta, and when infected, macrophages when infected with Neospora, we know that it's active IL-1 beta, not only pro-IL-1 beta, and that's induced only with live parasites and not antigens or attenuated parasites, only live infective parasites that are able to induce that IL-1 beta. Of course, if there's a IL-1 beta active, there's also a caspase activated. So when we infected those cells with parasites, we would see some activation of caspase 1 and 11, and as expected, there was a reduction in pore formation in the absence of caspase 1 and 11. But also strikingly to us that there was a very noticeable reduction of ROS production in the caspase 1 knockout cells. We went on to check on that, and we saw that actually in the absence of ROS, we couldn't see the activation of the inflammasome, and the other way around, right? Which was very striking to us that there's an active interplay between ROS production and the inflammasome to control the parasites. In the absence of those, you have increased parasite burden compared to wild-type mice, right? In knockout, different in knockout mice, cells of knockout mice, of different players of the inflammasome pathway. Right, so we also looked into the other side of the NOD-like receptors, NOD1 and NOD2. We actually found a phenotype on NOD2 mice. They had a lot less inflammation here in pancreas or in the lungs, liver, central nervous system. They had a lot less inflammation than wild-type mice. However, they had a lot more parasites, right, in the tissues. We did check, and we saw that they had less IL-6 and TNF-alpha in their samples in the knockout mice, which was also striking to us. And I think the most striking results to us regarding these experiments is that the NOD2 mice, they would survive more if dose that would kill or be lethal to 50% of the mice, or in a lethal dose to 100% of the mice. The NOD2 mice would survive the challenge better, which denotes to us that NOD2 actually contributes to the inflammation during the infection to rescue or to reduce the parasite burden, but this inflammation is sort of deleterious to the animal, and it actually causes immunopathology that kills mice, right? So was very, very interesting to us to see how this receptor can be targeted at certain moments of the infection, especially in dogs, right? We did also took a look at other receptors, now a different class, a C-type lectin receptors, or CLRs. We looked at dectin-1. We saw that dectin-1 mice were also more resistant. The dectin-1 knockout mice were also more resistant to the infection. However, in this case, they had less parasites, right? So we went on to see what was happening, and we were able to block dectin-1 with an antagonist. We would see less inflammation in tissues. We will also see increased IL-12 in these fluids or tissues, showing that the mechanism was kind of different from what we see on NOD2. The mechanism of protection induced actually that we saw in NOD2 mice was not real here. It seems that the parasite uses dectin-1 to sort of modulate negatively the response. It uses it to evade, and this does also have to do with ROS, right? In the absence of dectin-1, the animals would produce more ROS, and this would also trigger IL-12 production, right? So it's a different mechanism here. It's actually the parasite seems to be using dectin-1 and as an evasion mechanism. All right, just a sec. We went on to take a look at type 1 interferons versus the parasites. As you guys also know, this is what we want as a type 1 interferon response. It's actually when we get infected by viruses, we have a very sharp increase of type 1 interferons, and that seems to control the parasite burden. It's usually succeeded by a peak of type 2 interferon, especially interferon gamma, that has more power to reduce the parasite burden. So ideally, in the end of a few days, we went through the peaks of both types of interferons and reduced the pathogen burden, right? The problem is when we have uncontrolled type 1 response that usually is not able to control by itself the parasite burden, and this leads usually to a cytokine storm that's very deleterious to the host. So we wanted to understand how these pathways or these cytokines and these pathways were related to the infection by Neospora and Toxo. We first started looking at the receptor for type 1 interferons itself, and just to try to pinpoint the main results now regarding these infections and this cytokine. What we saw, as expected, that in the absence of the receptor, mice failed to control the parasites. It would be lethal. The infection would be lethal to these mice. They were not able to control the parasite burden in the different phases of the infection and in different tissues. Also, regarding inflammatory lesions, we saw necrosis, especially in the liver and in the brain. And this was also related to type 1 Th1 cytokines being produced in different tissues. So it was less in the absence of the type 1 interferon receptor. We would have less IL-12 and less interferon gamma in different sites of infection. And that would lead to, as also expected, reduction of nitric oxide, lack of parasite control, and death, right? So it all closes to where we expected. But what how does the parasite trigger the type 1 response, right? Type 1 interferon response. We first looked at the TLR3, that it's the TLR that does not signal through MYD88, and checked whether it had a role in this. We did transfect some macrophages with a reporter for TLR in the lab, which is green here. So when the parasite infects the macrophages, it does form a punta with TLR3, and this is not seen in toxo infection, right? It's not, we don't see this TLR3 punta in the toxo infected cells, which is a different trend, right? We saw that RF3, we had the same phenotype when the cells were infected with Neospora, which is the red parasites here. We would see a translocation of IRF3 to the nucleus of those cells, and that was not true for toxo-infected cells. In the absence of TRIF, mice would be more susceptible to infection by Neospora in different phases of the infection and different tissues. It will also, as we also expected, mess up Th1 cytokines, so less Th1 cytokines in different tissues, and also in gamma, right? Well, that was it for TLR3. Of course, STING is being assessed for different infections and different pathogens. We also did the same kind of experiments with transfected cells with STING GFP, overexpressing STING, and infected those cells. And in the presence of the parasite, we would have a higher expression of STING, or with the same readout for C-gas, right? So in the absence of in cells that were infected with Neospora, we would have increased expression of C-gas and STING. If we infected or incubated those cells only with Neospora DNA, we will also see increased expression of STING, right? So it seems that the parasite's DNA is activating STING. And one data that I didn't show to you guys about TLR3 is that when we pulse the cells with parasite RNA, we would see sort of the same phenotype. So it seems that parasite DNA and RNA induce type 1 interferons, and this protects the cells against the infection, right? So here we got some sting knockout macrophages that we did in the lab using CRISPR, and the cells are a lot more parasitized, right, after a short 24-hour incubation. Just to finish up my talk, I wanted to show to you guys what we're doing right now. We're doing some CRISPR in parasites. I did show to you guys some CRISPR in cells prior to this. Just to contextualize, Toxoplasma and Neospora, they have effector molecules that they produce and that gets thrown out in the cytoplasm or the nucleus of the cell, and they are able to modulate positively or negatively different pathways within the cell. And they want to enable to, one, multiply within the cell for freely, or two, to make the host survive the infectious challenge. And why would the parasite want the host to survive? As I told you guys before, these parasites keep latent in the brain for the rest of the lives of the infected hosts, and they pass the infection on to their predator, right? Of course, if you think about humans, we don't have a lot of carnivores that eat humans, but that's not true for all the other animals that we have around, right? So the parasites do secrete some molecules that are able to phosphorylate STATs, inhibiting IL-12, or inhibiting other pathways like the type 1 interferon pathway, inhibiting the cell to respond, okay, properly. They're modulating the cells as they wish. As I told you guys, we have used a lot of CRISPR in the lab to do genomic editing in parasites or in cells. For parasites, there are a few of these proteins called GRAS, which are dense granule proteins. They're proteins that are secreted inside the vacuole or in the parasite, in the wall of the parasitophilus vacuole, and they are used for the parasites to make their niche within the host cells, and also to block the cellular signaling pathways. For Toxoplasma, these mechanisms or this genome editing has been done for a while already, but for Neospora, no. We did actually a few years back develop tools to do this with Neospora, and we're taking now advantage of it to study the parasites. We did discover some GRAS in Neospora, that these parasites secrete effectors inside the vacuoles. We've described those. We know that a lot of those GRAS, when we look in Toxoplasma and Neospora, they're almost identical. A lot of their synteny, right? Almost all are common within the parasites. We wanted to study them and see if those parasite proteins are actually, how do they interact with the host and the host immune system in both parasites. We did some proteomic analysis using bioID to biotinylate inside the vacuole and then the vacuole wall to find a lot of those effectors. And we for the results I'm going to show you here to finish off my talk, we chose those two of those proteins. One protein that seemed to be upregulated in the chronic phase of the infection, and one of the proteins that seem to be upregulated in the acute phase of the infection. We took advantage of someone else's data set that was published at the ToxoDB site, and we went from here, right? We actually did identify those proteins by endogenous tagging by CRISPR in Toxoplasma and Neospora. We could see that those proteins exist in both parasites. Here we're looking at this GRA48 in both parasites. We also took a look at the one which is rich in the acute phase, which is also produced in Neospora or in Toxo. In Toxo, it seems to be a bit dim, but it's also in there, right? And we wanted to see how the host would behave in the absence of those proteins. So we set up an assay in the lab where we infected fibroblasts with Toxoplasma or Neospora and pulsed these cells for 4 days with a nitric oxide donor, which is sodium nitroprusside. Okay. After that, we would look if the parasites turned green, is that the parasites would go into their latent stage because of the nitric oxide pulse. And we wanted to check if those proteins were also expressed in both sides of the different stages of the parasites. So when we looked at graph 48, we have 2 vacuoles here. One big one that was an active vacuole that's full of taxoids, taxoids that are actually active forms of the parasites, the forms that are present in the acute phase of the infection. With everything that's red here is actually the epitope tag of the protein. So this big vacuole here is expressing the protein. This small vacuole here is also expressing, but look, it's green, so it's a latent stage phase, and we only see in the border. So, but the protein that we're looking at, graph 48, it's being expressed in the latent stage, the green stage, or the red stage. And that was not true for the other one, the acute phase protein. So that actually confirmed the RNA-seq study that we looked at. The green parasites, they do not have red or they do not have the GRA62 expression, but the red parasites, which are not green, right? It's being expressed. So this confirms that chronic stage parasites do express GRA48 active or not. And here for graph 62, only active tachyzoites would express it, not bradyzoites, the latent stage form. And what does that mean regarding infections of immune cells, right? So we got some of those parasites and we infected some macrophages and looked for cytokine production, especially IL-12, right? And IL-10 and IL-1 beta. We couldn't see a lot of difference between the wild type and the actual knockouts, at least for these 3 cytokines. But we did check for ROS production at the same experimental setup, and then we saw a big difference here for the acute phase protein. So in the absence for parasites that are knocked out of the GRA62, the acute phase infection GRA, a lot of the cells would produce a lot more ROS if compared to the wild type controls. Does that reflect anything on in vivo infection? So we looked at the chronic phase for the green parasites in the brain. We infected the mice, waited for 30 days, and looked in the brain for the green parasites. And what we saw is that those parasites that are knocked out of the acute phase protein that dampens ROS, actually those mice would have less parasite burden in the brain. Okay, so that's what we're doing right now. We're CRISPRing out cells and parasites and putting the model together to compare Toxoplasma and Neospora and the different factors. I think genomic editing has helped a lot our case with those possibilities we have with the cells and parasites, and we hope to be speeding up research towards finding immunobiologicals or therapeutics that can be effective against both infections. Of course, I have to thank a lot of the collaborators, students, or collaborators that helped us with this work, and of course also the financial support. And thank you very much.
Thank you very much, Tiago. It's amazing to see all the work that you have developed on modulation of innate immunity by those parasites. And I'm pretty sure people here have some questions for you. So we would like to start with students. You can ask or you can write the questions for us in the chat and we can read to Tiago. Otherwise, I have a question to start, actually. I was curious about the type 1 interferon response, right? So do Neospora and Toxo have different patterns of induction of type 1 interferon response, or is it? So Neospora induces and Toxo doesn't, right? Am I right?
Yeah,
Or
sort of, Diego. By TLR3, IRF3 pathway, it doesn't seem to use toxo any, right? So
Toxo doesn't induce the
doesn't.
toll-like receptor 3 response,
Yeah,
right?
yeah, exactly. So it does. We know that it does have a conserved induction by sting and cGAS. We can get type 1 interferon through that pathway in Toxo and Neospora, but it seems that only Neospora triggers the TLR3 pathway. It's something on its RNA that does it, and we don't actually know why this happened, Diego. One, I had a project with the student that did that work, most of the work that. We wanted to look for the motifs inside the parasite's RNA that would
Right.
trigger it. Some people have already questioned me if it's not like a retrovirus that is present in the parasite's genome or whatever. It's incorporated inside. Some people have looked for that, of course, in Leishmania, right?
Yeah.
Maybe we should check that and see if we don't have a retrovirus that it's inserted there, and we haven't seen it yet.
And if you get even our knockout mice, do they present different susceptibilities to Neospora and Toxo or not?
Well, for Neospora, it's very easy to see the
Right.
difference. For Toxoplasma, the problem is that it depends on the strain. It's so virulent that the differences between susceptibility between the mouse strains is not perceptible, right? So I can't answer that to you with a lot of precision because the experiments we did, the mortality curves were too close to each other, and I'm guessing that for Toxo it's sort of also true, but I mean I couldn't affirm by our results.
Thank you. So do we have more questions? Gustavo?
There's a question from Carolina. Carolina, if you can just open up the mic and ask yourself. If you have trouble, then we can meet it. But I'd rather have everybody asking alive. Carolina, are you around?
Do you want me to read it here, Gustavo?
I'll just read then.
Yeah.
The is really bad. Okay, just one thing, because I received a message in my WhatsApp. You guys need to plan ahead and get computer that is compatible, internet compatible. I don't care. It's a planning thing. You guys know that every Monday you need to get prepared to open up the video and ask questions. So I will talk to Anderson and Sergio, and we're gonna have a minimum of questioning. If you don't attain that number of questions during the semester, your concept will start being B, from A to B to C, and lose, the chance to get money from CCP. Okay, so I hope you guys understand. This is not a game. This is a serious thing. Okay, you can ask.
Carolina, Yeah, sorry, Gustavo. Carolina asked if I checked LC3, which is an autophagy marker, right? For in those cells. No, we did not check that, Carolina. We should actually do some autophagy work. I have to be truthful to you. I've been very resistant to look at autophagy, but I think I need to. We're doing some pyroptosis work right now to deepen that, but I should look at autophagy later on. A good question. I think we have Ruth also has raised her hand.
Yeah.
There's people have to close their.
Yeah, now I have to go ahead. I'll say that was a wonderful presentation. Actually, my question is not a question per se, it's just a form of curiosity. You pointed out that TLR3 is observed in Neospora caninum and not prominently in Toxoplasma gondii. What could have explained these differences? Because these two parasites are closely related. So what do you think caused these differences in Neospora having the TLR3 and not Toxo?
Yeah, so it's sort of where the same ballpark that Diego asked, right? So we know that parasite DNA, its recognition for parasite DNA from Toxo and Neospora is sort of the same, but RNA seems to be different, right? And especially through TLR3. We have not checked RIG-I or other innate receptors that also recognize RNA. So we should do that before we draw conclusions. But it seems something. What can I can affirm to you is that there's something in the parasite transcriptome that activates directly TLR3. I would guess it might be a virus, but I'm not so certain because people have done this with different strains of Neospora, and it seems to have the common phenotype. So I would not guess that it's likely that every Neospora strain has the same retrovirus, but it's formally possible. So we should check.
Oh, thank you for the well-detailed answer.
Tiago, I have just one quick question as well. We talked a lot about the innate immunity, but what's the relative contribution of CD8 cells to these two infections?
Great question, Gustavo. It's I had this full professor thing the end of last year, and Valderis was examining me, right? And she said, you just look at innate cells. You should look at T cells, right? And TCD8 cells are very important for the controlled infection. It's the parasite outside of the cell. It's very vulnerable, so it stays inside of the cell most of the time. And as you know, you need TCD8 to kill antigen coverage, right? The cells that are expressing antigens. So it's crucial. Without CD8, the host just simply dies, right? It's a complete susceptible phenotype. We know, which is funny for Toxoplasma at least. We know that around 70% of the CDA cells are actually raised against a single epitope of a single protein of the parasite. So we think it's like a decoy response. The parasite, induces a lot of that response, which gets it hurt, but it doesn't kill it, right? So we think that we do now have a parasite that it's knockout for that gene. And I really wanted to partner up with someone that do like the trimer work with the TCD8s and just to try to pinpoint other players, right? That might be hidden by this major response.
Wonderful. Thank you very much.
So, Tiago, I have another question that is more of a curiosity, but I think Anna has another question before me. So I already asked, so let's let Anna ask first, right? Ana Paula.
It's super quick. Thank you for the seminar. It was really. I was actually really curious when you mentioned I think the parasite upregulating sting was activation.
We saw upregulation, right?
Upregulation of the protein. That was quite interesting. Do you have any insight of how it's being upregulated in the cells?
Well, I would guess if it was only during the active infection, it was because of the parasite's, replication status or whatever inside the cell. But that also happens when we pulse the cells with DNA only with like liposomes, right? Incorporated into liposomes. So I don't know, Ana Paula. It's weird, right? These parasites, they do a lot of stuff to keep the host alive. They don't want the host to die, and I would guess something is triggering it up so they can produce some inflammation. But it's just a wild guess. I wouldn't know how or why this is happening.
Okay, all right, thank you.
Tiago, just a philosophical thing here, because you just mentioned that. So, do you believe the parasite is keeping the host alive, or is just not killing the host and reaching a? So, is there anything active that may you think that what's the
they actually actively induce inflammation, Gustavo. There's one of those secreted factors that binds directly to TREF6 to induce IL-12 and induce gamma to keep the parasite burden down, right? Because you can't kill it. It's gonna travel all over regardless. It uses like monocytes as Trojan horses, so it goes to the brain regardless, and it wants to be there at latent stage to be, when a cat eats a mouse, it starts by the head, right? And the parasite is there. So the parasite's swollen, and by the cat eliminates the environment stages, and the cycle goes from there. So we know that actively induces survival of the host.
Right. So that's exactly my point. So you think that this is a response that evolved from the parasite? It's not like the host trying to, interact.
No, I think it's something that evolved from the parasites. Because we have a strain of Toxoplasma which is very virulent to mice. A single parasite kills a mouse. So, and that parasite, we know that it does not have those factors that keep the host alive. It just has the ones that turn it off, right? The ones that phosphorylates that 6 and 3 and blocks all the other mechanisms. So it's so virulent because it just turns off and doesn't turn anything on.
That's very cool. Thank you. Anybody else?
I had a question out of curiosity, actually, Tiago. So when you were mentioning about toll-like receptor 11 and 12 and profiling,
Yeah.
right? So Neospora also expresses profiling, right? It also
Yes.
triggers toll-like receptor 11 and 12. Do dogs express toll 11 and 12?
Oh, good question. Good question. I don't know. Good question. I'm gonna look now and let you know. But I don't know. I don't know.
Because humans don't, right?
Humans don't.
This was a huge question mark. How does toxo activate tolls in humans if the main molecule triggers something that they don't have?
Exactly. So I don't know if you remember. I don't know if you were present last semester. Melissa Lodoan from UC Irvine, and her line of research nowadays is that's the exact question. If humans do not have active or do not have in the genome TLR11 and 12, how do they respond, right? And it's a bunch of different mechanisms. A lot of inflammasome, a lot of type 1 interferon. that does the same part. It seems that in the mouse, TLR 11 and 12, they actually evolved to control toxo, right? It's a huge part of the. So yeah, good question, Dave. I don't know. I'm gonna look for it right now, and I'll let you know in a few minutes.
All right. Thank you, man. So do we have extra questions here, guys?
Okay,
I think we're done.
then we done. You guys
Obrigado.
enjoy your week. Remember, for those who arrive late, that next week we will have the seminar at the Teatro Rosa here. So I look forward to meet all of you. If by any chance you have something on that day and you need to go remotely, let us know in advance. Find a place with good internet and video camera, and be prepared to ask questions. This is the job, really. The sitting in front of the screen watching is very passive. Okay, so you be more active. The learning is to ask questions. So be prepared to ask questions. All right, guys. Tiago, namaste. Thank you very much. You guys enjoy the week. Bye.
Thank you.
Bye-bye.
Obrigada, Tiago.
Obrigado.
Obrigada,
Tchau,
Diego.
gente.
Gustavo.